While major depressive disorder can affect anyone regardless of gender, research has shown that men and women may experience depression differently. Women are twice as likely to suffer from depression as men and have a different subset of symptoms which are often more severe. This affects treatment as women respond differently to antidepressants compared to men. While researchers have acknowledged the differences of major depressive disorder in men and women, we have yet to uncover the molecular reasons for this.
Today, we may have an answer. For our podcast today, we have the privilege of chatting with Dr. Eric Nestler, Director of the Friedman Brain Institute and Chief Officer of the Mount Sinai Health System. In December 2022, he published a paper titled The Long Non-Coding RNA Fedora is a Cell Type and Sex-Specific Regulator of Depression. This paper discussed the implications of a long non-coding RNA nicknamed Fedora in regulating depression-like behavior within female mice.
We're excited to learn more about this research and what it means for the treatment of depression in the future. So to start off, your paper, the effects of Fedora in regulating depression-like behavior within female mice was really interesting. And I guess to start off, I would love to know what led you to investigate the Fedora gene as a potential regulator of depression.
Yeah, absolutely. So we had done a study on taking postmortem brains from people who died, right? And half of the people were depressed when they died, half of the people were normal, not depressed when they died. This is part of a ghoulish practice of having brain collections around the country. Because unlike other organs, this is important for you guys to appreciate. You know, heart, lung, liver, kidney, you do a biopsy of your patient. No one is going to do a brain biopsy of someone who's depressed, right? You just can't do it. It's not safe. So we have to wait for a person to die and then study their brains. So then we used a technique called RNA sequencing, which is a method that quantifies all RNAs present in a tissue. Maybe there's 20, 30,000 or so different RNAs present in every tissue in the body. So RNA sequencing gives you just a table with a value, the level of every one of these 20, 30,000 RNAs, right? And you get that list from people who aren't depressed and people who are depressed. When you go through those lists of RNAs, you realize that only about half of the RNAs are actually encoding proteins. Well, it turns out that about half the RNAs present in the brain don't make proteins. They're called non-coding RNAs. And these were unknown to the world up until a few years ago. A postdoc in my lab, Orna Isler, first author of the paper, took these data and she started analyzing these non-coding RNAs. And she found some interesting things. First, just in terms of basic biology, the percent of all RNAs that are non-coding has increased the more recent we get in evolution. So if you look at an invertebrate animal, for example, a large majority of its RNAs code proteins. When you get to monkeys and humans, a majority do not encode proteins. So that's interesting. There has been an especial expansion of long non-coding RNAs in primates. So not only mammals, but in primates. And about about a third of all of those long non-coding RNAs are enriched in brain. This led to the idea that, gee, these may be really important for what makes humans special. The next thing Orna noticed was that there were tremendous differences in levels of expression in the brains of women versus the brains of men. And then what she found is then if you look at the people who are depressed, there was even bigger differences in the long non-coding RNAs that are abnormally expressed in depression in women, not so much in men. So here was this very specific and dramatic difference in a gene expression abnormality seen in depression in women that was not seen in depression in men. And that prompted Orna to look more deeply into the specific RNAs that she studied.
I think that's like really interesting that
Yes.
your postdoc like managed to analyze everything and figure out this pattern, which helped you
Yep.
guys in this paper.
In your paper, you mentioned that Fedora regulates depression in a cell type and sex-specific manner. Could
Mm.
you elaborate on what this means and why it's significant?
So what Orna did once she got this list of RNAs, that these long non-coding RNAs that are differently affected in depression in men versus women, she ranked them based on their, degree of difference, degree of regulation. And this long non-coding RNA that she ended up calling Fedora was top ranked. So the next thing she did was to go online and ask, based on work that other people have done, what cell type in brain expresses Fedora. Half of the cells in the brain are not nerve cells, they're called glia, right? So there are different types of glia. There are oligodendrocytes, microglia, and astrocytes, and then also blood vessels. And it turned out that Fedora was most expressed in two of those cell types, nerve cells and oligodendrocytes. And that prompted Orna to look specifically at actions in both of those cells. Oligodendrocytes provide myelin for nerve cells. Have you heard of myelin?
Yes.
Okay, so every nerve cell has a big, juicy cell body, like every other cell. And what makes neurons different is that they send processes to other parts of the brain, right? They have the branches of a tree called dendrites that receive inputs, and then they send out axons that send signals to other nerve cells' dendrites. The only way that axons can send those signals fast and across long distances in a human brain is if the axons are myelinated. Myelin is simply a series of plasma membrane that gets wrapped around every axon. So oligodendrocytes are sitting there. I'm trying to think of what this might look like. Oligodendrocytes are like an octopus that sends its tentacles wrapped around axons. And the tentacles that are wrapped around the axons are just simply membrane of the oligodendrocyte that insulates the axon. Orna found that this Fedora was expressed in nerve cells and in oligodendrocytes. And then she went ahead and studied what activity Fedora exerts in both those cell types. In the nerve cells, what she found is that it controls the electrical activity in nerve cells. That was published in the paper. In oligodendrocytes don't have electricity, but they make this myelin, and it turned out that Fedora altered the amount of myelin around the axons. So here's the same long non-coding RNA expressed in two different cell types, nerve cells, oligodendrocytes, and in each cell type, it selectively regulates the functioning of that cell type, electricity in the nerve cell and myelin in the oligodendrocyte. But it only does it in females, not in males.
That kind of bounces off one of the questions I had. I was going to ask, could you expand on how Fedora expression in oligodendrocytes causes the thinning of myelin? So that pretty much answers my question to
Yeah,
that.
we don't know. Presumably, long non-coding RNAs work generally by altering the expression of protein coding genes. So we think that Fedora is in oligodendrocytes, regulates genes that control myelin thickness, whereas in nerve cells, Fedora controls genes regulating the electrical activity of nerve cells, and that's something that we're studying now.
In the paper, you guys mentioned that you guys studied it within different mice, female and male mice. And, I would like to know a little bit like, what about the challenges that were presented, when studying with mice? For example, like there were a series of behavioral tests and like the elevated maze, I think. And my question is, how do you know like a mouse exhibits truly like depression like symptoms rather than being like, oh, I don't like the mice doesn't just want to do that specific action?
Right, so that's a really critical question and gets at the heart of what we do. Ultimately, I would say it's impossible to know if a mouse is depressed or anxious, right? Because the only way that we know if a person is depressed or anxious is to talk to the person. Since mice don't talk to us, there's no way to know. So the goal of the field is to try to develop tests that will provide some insight into that question, understanding that perfect answers are not available. The inference is that when an animal ventures out less, when we overexpress Fedora, our hypothesis is that the animal ventures out less because it's feeling more fearful.
That makes a lot of sense. I think it's interesting how you're saying all mammals are curious. That makes a lot of sense for this
Yeah,
paper.
yeah. And it really is interesting. I don't know if either of you have had experiences with pet dogs or cats, but you really do get a sense of how that curiosity is something that really is a mammalian trait. But the fact that Fedora was found in depressed humans, an abnormality in expression levels of Fedora was found in depressed humans, gives us confidence that we're looking at something real and significant in the mouse.
And leading on to our next question, your paper suggests that targeting Fedora could be a potential therapeutic strategy for treating depression.
Mm-hmm.
What are some potential challenges or limitations to this approach?
Right. So the challenge is, like with anything in the brain, is I am not going to want to drill a hole in somebody's head and inject something into their brains, right? So the challenge is always, what can we give somebody by mouth or in their bloodstream with an injection that would affect Fedora levels in their brains? We would want to put in something in that lipid nanoparticle that would then get into the brain and block Fedora action.
That's actually very, interesting to hear about. And I guess perhaps that would be mostly like the next steps for this experiment.
Yes, absolutely.
I did have a question about your paper states that singling out the role of single RNA in a disease as complex as major depressive disorder presents many challenges, including the fact that mice formally do not express Fedora. So, could you still explain about how mice were still dependable in the experiment, given this fact? I think you talked about it
Yes.
before, but
Yeah, so here's a good question. So here we are in the lab a few years ago. We find Fedora robustly regulated only in women with depression. Amazing finding. Okay, now what? What do you do? How do you study it? Fedora is not expressed in a mouse or a rat or any other animal other than a monkey. What Orna and I've conjectured is that, okay, long non-coding RNAs typically work by binding to something based on their structure, right? Their structure gives them specificity that enables them to bind to something. Just like a hormone binds to a receptor, right? There's specificity in the structure of those two molecules that makes them bind together. And we figured that even if Fedora isn't expressed in a mouse, there's a decent chance that some of the things to which Fedora binds to in human brain are already present in mouse brain. Almost every protein in a human is present in a mouse. For example, a chimpanzee and a human are 98% the same when it comes to protein coding genes.
May I ask if Fedora is present in monkeys? Why would you not attempt to test on them?
Ultimately, Isabel, it would be valuable to do so, but the regulatory burden and expense of studying monkeys today is extreme. and it makes the research extremely difficult to do.
Makes sense.
But it's really, that is the main reason why not. And if we're thinking of injecting nanoparticles with a Fedora inhibitor as a treatment for depression, that treatment is going to have to go into monkeys before it goes into people for safety reasons.
The long process.
Yes.
Another question we were wondering about was how ketamine works for patients who suffer with depression and how that interacts with Fedora.
Yes. So ketamine, as you may know, is a new treatment for depression. Ketamine is very different than all other antidepressants because when we give ketamine to a person, their depression resolves within hours. Fedora and ketamine seem to be somewhat interacting though, because when we did look at human blood in that study that you are looking at, we found that Fedora levels were abnormal in the blood of depressed patients. So this is an example of a finding in blood that's reflective of an abnormality that's present in the brains of these people. And then when the people were given ketamine and got better, the Fedora levels normalized.
That's actually a great segue into my next question, which is, what are some of the next steps in your research on Fedora and its role in depression? So you mentioned you kind of want to get that idea of like, hey, just take a blood test, and I'll see, if Fedora levels are elevated. So what are some steps would you have to take to reach that idea?
The main step is to just do that experiment instead of, I forget the number of people we looked at in this paper, maybe 20 people, I forget. We'd have to look at 2,000 people. So in order to develop a new blood test, you need to look at thousands of humans. So this is what we're in the process of trying to do, just really massively expand the study and see if it provides a valuable readout or not.
For you, what would be like a valuable readout?
Well, at this step, it would be to see whether a person before treatment who's depressed has elevated Fedora levels. And the prediction is that when a person is treated and gets better, their Fedora levels normalize. Whereas those people who are treated and don't get better, their Fedora levels do not normalize. And that could be useful because oftentimes when I treat a person who is depressed and they take an antidepressant medication and they get better, life is complicated, right? Bad shit happens to people. And a person might have a relationship end, they may get a bad grade in something, a parent gets sick, whatever it is, right? Life intervenes. And then they feel sad again. Their biggest question when they call me up frantic and panicking is, oh my God, am I getting depressed again?
When you mentioned normal sadness versus clinical depression,
Mm-hmm.
do you believe that, say, Amina and I get really sad one day, we get a bad grade on this.
Yeah.
Do Fedora levels increase, or is that only with clinical depression?
We don't know, and that's what we're going to have. That would be part of this study as well. Precisely.
Okay.
That's exactly what it is.
Well, thank you so much for having this interview
It is
with
my
us today.
pleasure, guys. It's really nice to meet you both, and good luck with your studies. I hope that you get an A as you deserve it, and let me know if I could help further.
Thank you so
Thank you so much.
Okay, all the best. Take care.